The DNA double helix is a chemically dynamic molecule that exists in a constantly hostile environment. From the inside, normal cellular metabolism continuously produces reactive oxygen species that attack the nucleotide bases. From the outside, environmental mutagens like ultraviolet and ionizing radiation deliver energetic strikes that cause the rigid structure to spontaneously kink and fracture. When these physical and chemical modifications are left unrepaired, the continuity of the genetic code is broken. The immediate cellular consequences are mutagenesis, a permanent arrest of the cell cycle known as senescence, and eventually uncontrolled oncogenesis. Maintaining genomic integrity requires an active, highly specialized enzymatic toolkit. The cell deploys specific repair machineries dictated entirely by the exact structural nature of the lesion it encounters. This hierarchical tree diagram maps the entire molecular landscape of DNA damage and repair. By categorizing the repair pathways based on their target lesions, we can see the exact physiological logic the cell uses to protect its genome. The taxonomy first divides into endogenous versus exogenous sources of damage, and then diverges into specific branches. We see direct reversal mechanisms, single-strand excision pathways like BER and NER, mismatch repair, complex double-strand break resolution, and finally damage tolerance. Success in rigorous academic assessments like the CSIR-NET depends on high-resolution knowledge of this map. This means internalizing precise. We begin with the most frequent internal threat. This graphic highlights a CSIR-NET question identifying depurination as the most common spontaneous DNA lesion. In the aqueous environment of the nucleus, spontaneous hydrolytic reactions sever the N-glycosidic bond, linking a purine to its sugar. This creates an apurinic site at a staggering rate of approximately 10,000 events per mammalian cell every single day. To address highly specific modifications, cells utilize direct reversal. This is the most energetically efficient category of repair because it strips the damaging modification away without cleaving the phosphodiester backbone or requiring a complementary template strand. This animation demonstrates the repair of 0,6-methylguanine by MGMT. MGMT flips the damaged base out of the helix, and an active site cysteine directly accepts the methyl group. This inactivates MGMT, targeting it for ubiquitin-mediated proteasomal degradation. As tested in the 2020 CSIR-NET, this confirms MGMT is not a true enzyme. It is a suicide protein, irreversibly inactivated after a single reaction. This breakdown of the June 2017 exam contrasts two other direct reversal proteins. While DNA photolyase requires blue light photons to repair UV dimers, ALK-B utilizes iron-2 and alpha-ketoglutarate to perform oxidative demethylation in a strictly light-independent thermochemical reaction, synthesizing an entire protein only to destroy it after removing a single methyl group incurs a massive thermodynamic penalty. The fact that the cell willingly pays this cost proves the extreme priority biological systems place on maintaining absolute genomic fidelity. When lesions do not massively distort the DNA helix, cells deploy base excision repair. This question isolates uracil DNA glycosylase as the specific initiator. These glycosylases constantly scan the minor and major grooves, locating non-bulky modifications like uracil and cleaving the base to leave an abasic site. This chronological flowchart maps the long patch sequence tested in the December 2024 exam. After the glycosylase removes the base, AP endonuclease 1 cleaves the phosphodiester backbone. If the resulting terminus is modified, DNA polymerases delta or epsilon synthesize a multinucleotide patch, displacing a flap that is subsequently cleaved by FEN1 and sealed by DNA ligase 1. Base excision repair is inadequate for large, structurally obstructive damage. For bulky lesions like UV-induced pyrimidine dimers, the cell shifts to nucleotide excision repair, or NER. This animation illustrates the prokaryotic UVR-ABCD system recognizing structural distortion. After the UVRA-UVRB complex binds the lesion, it recruits the UVRC endonuclease. UVRC makes highly precise dual incisions, exactly 8 bonds 5' and 4 to 5 bonds 3' to the lesion, excising a clean 12 nucleotide fragment. The eukaryotic equivalent, transcription-coupled NER, utilizes a completely different sensor. RNA polymerase II actively transcribes the DNA until it physically stalls at the bulky lesion. This stalling serves as the primary sensor, immediately recruiting Cockayne syndrome proteins CSA and CSB to initiate repair. The exact physical bulk and geometric distortion of the mutation rigidly dictate the mechanism. A single unnatural base triggers a localized cut and patch response, while a rigid kink in the double helix forces the cell to excise an entire segment of the strand. Even high-fidelity replication polymerases make errors. Mismatch repair rectifies these mispaired bases, but it faces a severe mechanistic challenge. It must perfectly differentiate the newly synthesized daughter strand containing the error from the original, correct parental template. This diagram shows how E. coli solves the strand discrimination problem through transient hemimethylation. DAM methylase applies a methyl group strictly to the adenine of a GATC sequence on the parental strand. Immediately post-replication, the nascent daughter strand lacks this modification. The repair machinery exploits this temporary state. The MUT-H endonuclease specifically binds and introduces a nick exclusively on the unmethylated strand, ensuring only the newly synthesized sequence is excised. Eukaryotes completely abandon methylation for this process. As shown in this July 2024 exam breakdown, eukaryotic MMR requires MUTS-alpha and MUT-alpha complexes. Instead of mu-TH, the intrinsic endonucleose activity of MUT-lel-alpha is stimulated by the pCNA sliding clamp, which naturally coordinates with the newly synthesized strand during replication. Reliable strand discrimination is the absolute prerequisite for mismatch repair. If the enzymatic machinery cannot identify the parental template, it would randomly guess, introducing exactly as many mutations as it attempts to fix. Double-strand breaks are profoundly cytotoxic, and the cell uses this cycle dial to govern its repair strategy. Homologous recombination strictly requires an intact sister chromatid as a template. Therefore, it only occurs in the S and G2 phases, where active CDKs phosphorylate the CTIP protein to initiate extensive end resection. This biochemical gatekeeping is a critical exam parameter. Without active CDKs, resection is blocked, completely restricting homologous recombination to the late cell cycle. During other phases, cells normally rely on non-homologous end joining, but if that pathway is compromised, this flow diagram shows the alternative, microhomology-mediated end joining. PARP1 recognizes the break, initiating a limited resection to expose short microhomologies, which are hastily fused by polymerase theta and ligase 3. When lesions completely stall the replication fork, the structural integrity of the entire chromosome is at risk. This triggers damage tolerance. In E. coli, large tracts of single-stranded DNA cause REC-A to polymerize into a nucleoprotein filament. REC-A acts as a co-protease, binding to the LEX-A repressor and inducing autocatalytic cleavage, de-repressing the massive SOS emergency response. In eukaryotes, translesion symphysis is managed through strict architectural modifications. Stalling replication forks trigger the precise monoubiquitination of the pCNA sliding clamp. This modification actively displaces high-fidelity enzymes, substituting them for specialized error-prone TLS polymerases that can power through the blockage. These tolerance mechanisms deliberately introduce errors. Confronted with unrepairable damage, the cell makes a calculated biological compromise. It chooses to incur a permanent mutation rather than suffer the immediate catastrophic lethality of a collapsed replication fork. The presence of unrepaired DNA serves as a physical distress signal. It activates the ATM and ATR kinase networks, triggering a global checkpoint response that forcibly arrests the cell cycle until the damage is resolved. Returning to our taxonomy tree, we map inherited mutations in these pathways directly to human pathology. A defect in core NER genes causes xeroderma pigmentosum. Failure in TCNER triggers Cockayne syndrome. Losing MMR capabilities drives massive microsatellite instability in Lynch syndrome. These devastating clinical phenotypes, ranging from extreme cancer susceptibility to premature neurodegeneration, are the macroscopic manifestations of microscopic enzymatic failures. Clinical pharmacology can actually exploit these genetic defects. Synthetic lethality is a therapeutic strategy that forces tumor cells to rely on repair pathways they physically do not possess. Consider breast and ovarian cancers carrying hereditary BRCA mutations. By administering PARP1 inhibitors, clinicians deliberately block base excision repair, causing single-strand breaks to persist. When an active replication fork collides with one of these trapped structures, it violently transforms a simple nick into a lethal double-strand break. As this clinical pathology question details, a healthy cell will easily fix this utilizing homologous recombination, but a BRCA mutant cancer cell lacks HR entirely. It cannot repair the resulting double-strand break, forcing it into targeted apoptotic death. While DNA damage invariably drives cellular aging, this graphic highlights a specific exception tested in the June 2025 exam. The transcriptional activation of telomerase in somatic cells adds hexameric repeats to chromosome ends. This actively mitigates senescence and counteracts the aging process. Genomic integrity is never guaranteed. It is actively enforced by a rigorously regulated, thermodynamically expensive network of sensors and effectors. Resolving how this system dictates pathway choice provides the fundamental framework for understanding both cellular evolution and modern targeted clinical pathology.